Hi, everyone. As you know, my name is Patricia Miller, and I'm a microbial ecologist. I specialize in microbiome profiling of plants, including grasses. And I also have research experience in lake and soil sediments, as well as bioremediation. And I specifically focus on microbiomes of plants, and lately, especially in the turf grass sector. So yeah, thank you so much for joining us. And today I just wanted to talk a little bit about cold season transition and how microbes and microbiomes of grasses and soils can contribute to that successful transition. So yeah, thanks again for joining me. I'm just going to go ahead and start here. Okay, so I divided the talk into two topics. So the first one, I'm going to give you some background information. And of course, the theme is the science behind. So it's going to be a little bit science intensive. And I'm going to be teaching you the latest research of the science behind winterkill. So what causes winterkill? What is the role of the grass and soil microbiome? And what types of cold stress damage occur? And then in the second half, we're going to talk a little bit about mitigation strategies and how we can use microbiome management to help smooth over that transition between winterkill effects and recovery. Okay, so part one, first going to discuss what is winterkill. So probably most of you know, probably saw some of you possibly at the OGSA conference earlier today. So you're going to know a little bit more about some of this topic than I am, but I'm going to focus more on what I'm seeing in literature in the academic sector. So what do academics, I guess, say about winterkill? So it generally describes turf losses occurring directly because of winter conditions. So there are two categories that you tend to see in the literature. You see abiotic and biotic stress. And the results of both are the same. You either get damaged or stressed grass, or you get dead patches of grass. Okay, so the two types, biotic deals with sort of live effects, so organisms. In this case, we're specifically going to be talking about fungi and things like snow mold, and I'm going to touch a little bit on nematodes. And the other type of stress is a more physical stress called abiotic stress. In our case, we're going to discuss cold stress, and we're going to talk about water deficits. But there's lots of other stresses that, of course, your grass is dealing with throughout the season. So here's some interesting pictures here. You see the effects of two types of biotic stress. So you've got low temperature diseases. So these are cold loving fungi, and they cause infections after a prolonged and heavy layer of snow. So if we get a lot of snow, and if you had snow in your area, then this is the often and sadly the effects. So the two common types of snow mold are gray snow mold and pink snow mold. Gray snow mold is caused by the organism Typhula incarnata. It tends to affect leaves. And so you can see it up here. You can see the effects of the leaves in this area. You can see the gray mycelium, which is like the parts of the fungi that are affecting the leaves. And this tends to affect more of the leaves and stems and not so much the grass crowns and roots. So you do get a little bit faster recovery if this is the type of mold affecting your grass. Pink snow mold, on the other hand, is a little bit more severe, and it tends to affect the roots and the crowns. So you end up with more damage and more overall loss. And you see this sort of interesting speckled pattern here. And if you look closely, this is what pink snow mold tends to look like. Very circular in the way that it attacks the grass. And again, you can see the mycelium, which is the fungus here on the outside. And the issue with this particular type of mold is you tend to get a fusarium patch in warmer temperatures as well. It tends to affect more close cut grass. So you'll see it more on putting greens, tees and fairways. And also it can affect recently seeded young fall grass. So if you seeded in the fall, you could see the effects in the spring if that fall grass didn't establish. And also spring fungicides tend to have a little or no impact according to the research. And but if you did spray in the fall, then you have a little bit better chances of avoiding that. Okay, so what are the abiotic or the physical stressors that winterkill tends to cause? So the first more severe type is ice encasement. So this tends to happen if on your golf course you have sort of these low-lying areas that are covered in ice. If you have those areas, then and this type of ice lasts for more than 30 days, you can get a buildup of toxic CO2, and this can be really devastating to your grass, and it can essentially suffocate the grass. The more common types of physical stressors that you'll see, though, is crown hydration and winter desiccation. So these are the sort of more common. You see here on the left, this is crown hydration, and then on the right is the desiccation. So crown hydration is when there's a sudden thaw in temperature, and then you get a sudden freeze after that. So during the thaw, the crown or the base of the grass is absorbing a lot of water. Once you hit about that 80% saturation, then that water will freeze and you'll get a sudden low temp. And then, of course, the grass will burst and die. The other thing that can happen is you can end up with ice crystals inside. And again, it's the same type of thing. You get break through the crown tissues. And again, you get these dead areas. The other type is called winter desiccation. So this is caused when you see higher winds and very little precipitation. And this essentially mimics drought effects. And so here on the right, you see a little bit different coloring here. And it's very similar to the type of drought problems you would see in the summer. And this tends to affect the grass roots as well as the blades. Okay, so here I just took a look and did a little bit of research on the mitigation strategies. Obviously, in the golf course industry, you're the experts on the mitigation strategies. I'm going to touch on the things that I know a little bit more about, but the things that you would know more about would be things like physical barriers and snow removal. So this is one way of doing that. You can also reduce fungi by debris reduction, so by removing thatch. And the reason is the thatch tends to have mold spores attached to it. So you just can rake those dead areas, kind of remove that, and that will reduce sort of that cycle from year to year. And then there was a little bit of research I saw on crown hydration prevention, and they were suggesting aeration and sand top dressing to minimize that. The areas that I'm going to focus more on this talk are going to be on overseeding. Research tends to prefer verticutting with overseeding, and the timing and success will depend on the disease rate. So when should you overseed if you're dealing with snow mold, for example, or other types of fungal infections? And of course, the other mitigation strategy that's becoming more and more popular and has more research dedicated in recent years is biostimulants and biofertilizers. So biostimulants and biofertilizers can also fix nitrogen, so supply nitrogen to your plant, can reduce stress, can also defend against pathogens and improve growth, as well as improve soil biodiversity. This is really important. So I'll touch on these topics in the next few slides. And products that you've probably seen before are obviously composts, humic acids, amino acid-rich formulations, and what we're going to focus more on is beneficial plant and soil bacteria. And fungi are also useful and can be very similar to bacteria. So what is the role of biostimulants and biofertilizers? So I did find an interesting paper that was written last year called Biostimulants in the Production of Forage Grasses and Turf Grasses. And the authors there did a full review. Here you can see all the different biostimulants that they reviewed, including beneficial bacteria. And what they found was there was a whole plant response. So you see all the different sort of benefits here. And I believe at the end, if you request that, we can give you a list of all of these references. So in this paper, one of the key things they talked about was that biostimulants enhance the quality of grasses, augments their tolerance to environmental stress, can facilitate nutrient uptake like nitrogen, phosphorus, and iron, and can also improve the visual quality of grasses. And they're not necessarily meant to replace fertilizers fully, but they can significantly improve crop effectiveness in utilizing the nutrients present in fertilizers, as well as increasing carbohydrate storage. And most importantly, they can also increase microbial activity and the biodiversity of both soil and plant microbiomes. And this is really important for improving your grass's defense against fungal pathogens, as well as the health of your turf. So what are the impacts of beneficial bacteria in plants as well as soil? Sort of what is their main functions? So I found this interesting article here, thought this was really cute. Some sort of like all the different benefits of what bacteria can provide and biostimulants. And what I did is summarize sort of the three main categories. So the first is that they produce phytohormones. So synthetic and organic amendments like humic acids or seaweed, they can stimulate phytohormones, but plant and soil beneficial bacteria are the ones that can actually produce them. And they don't just produce them one time, but they produce them continually while growing with the plant. So this is what distinguishes beneficial bacteria from other sort of amendments that you might use. They're also involved in decomposition, nutrient cycling, soil structuring, and of course, we talked about abiotic and biotic stress regulation, so they can help deal with that as well. They can enhance the effectiveness of a synthetic. So this is something that I'm seeing a lot of in research in the last 5 years, that they are actually able to extend the length of your synthetics and extend how long that you can wait between each application. And this improves plant and soil health while enhancing that effectiveness. And then the final category is plant immunity. So not just plant immunity, but also soil immunity, or something called soil disease suppressiveness. So adding beneficial bacteria can activate the plant immune system, and it does this by stimulating specific hormones like jasmonic acid, and this initiates something called the induced systemic resistance, or basically the immune system of the plant. Another way that it helps reduce pathogens is by scavenging iron away from pathogens. And also specific beneficial bacteria that can colonize both soils and plants, add diversity and resistance to soil disease suppressiveness. And they do this while at the same time stimulating roots and the rhizosphere. So it has benefits for soil, plants, and your roots. Okay, so here I just put a picture where you can see the good soil, the bad soil, and this is just sort of a depiction of what happens between soil and plants. So plants and soil microbiomes do something that's called crosstalk, so they can speak to each other. And the way they do that is they send signals to each other. And what I found in the research when I looked through is sort of the theme in the last 4 years has been that ecosystem management has strong impacts on the biodiversity. So that's why here they've got the good soil, the bad soil, and it can reduce soil bacterial diversity. It can also decrease the availability of available carbon and nitrogen. But the good news is that when they examined specifically turf grass systems, they found that though you might have a lower carbon and nitrogen, they did not find less diversity. So that means that there are still good bacteria in your soil, and we have to find ways to draw those out. The other theme that I'm seeing regarding microbiomes is that agrochemical use has increased fungal and bacterial infections due to a reduction in the natural soil disease suppressiveness. So we talked about that in the other slide. And what's happening is you see an overall decrease in grass yields. And this is because turfgrass diseases tend to be caused by fungi, and agrochemical use is sort of encouraging fungal pathogens. It can also lead to this term called dysbiosis of microbiomes, sort of like an imbalance of microbiomes in soils and plants. And also domestication. So studies have found that domestication has also disrupted some essential plant-soil interactions. So when researchers look at wild types, or we'll call them ancient types of systems, then they're able to see that domestication has reduced a little bit of that biodiversity. And the good news is that microbiome reinforcement has been shown to be able to fill that gap. So we're going to talk a little bit more about that today. Okay, so we're already into part 2. So here I'm going to talk a little bit more about microbiome management. So how can we manage winter kill recovery and manage disease stress? So abiotic stress like winter desiccation, so this is drought. I'm also going to talk about nitrogen, about the importance of balancing not just nitrogen fertilizers, but fertilizers in general, and balancing these inputs to help reduce pathogenic fungi, as well as improving soil diversity and function, and ways to do that without compromising available nutrients to your plants and your soil. And then thirdly, I'm going to discuss how to reduce fungal diseases, including snow molds, in order to allow for overseeding of damaged zones a little bit earlier. Okay, so the most important thing that I saw in literature and what I keep seeing is, and something I'm sure you know, is that root architecture, or the length of your roots, is the most important thing for winter desiccation, along with nutrient storage. So the longer the roots, the more root structure you have, the healthier your grass is, the more nutrients it can reach, and the better it will be able to withstand stress. So when you have longer roots, water can be reached, and it helps mitigate drought-like conditions. And this is facilitated specifically by phytohormones, which we know that both plants and bacteria can produce. This helps plants grow those roots to access nutrients it wouldn't otherwise be able to reach. There was a study in 2022 that I found interesting about sort of the drought mechanism. So how is your grass dealing with drought? And one of the ways that they found it can deal with it is by doing this interplay between two hormones called auxins and cytokinins. And what it does is it regulates your plant growth. So when the plant detects drought, then it signals itself to close the stomata and reduce shoot growth and favor root growth, as well as architecture. And this maximizes water and nutrient retention. This is really important for dealing with drought. And in 2020, they found similar results in tall fescue as well. And then in 2023, another research group found that when they used inoculants, bacterial inoculants in creeping bentgrass during drought, that they were able to significantly increase tillers, increase root viability, and of course, root growth and length. So here in the graph, I'm showing here in black, you see the controls. So this is well watered. So this is before drought. You can see that the inoculated grass gave a lot more total root length. So this is important prior to drought conditions so that your grass can establish the roots and withstand the drought. But even if you're dealing with a post-drought situation, here you see after it was re-watered and inoculated, again, you see a significant difference. That's what the star here means after the drought. So they watered 15 days after a 35-day drought, and they still saw the significant difference. And they saw this also in an increase in tillers and other parameters as well. So inoculants can improve the root structure both before as well as after drought. Okay, and if you have any questions, just add those into the chat. And at the end of this, I'll be able to answer those. So that was already a lot, and I still have just a few more slides here. So what are strategies that you can do in regards to fertilizers to help deal with winter stress? So we know that nitrogen in literature, that it's the #1 limiting nutrient in turfgrass systems. Of course, I'm sure your experience points to that as well. And it is the key for winter kill mitigation and root architecture increases. So improving nitrogen use in the grass is most important. And there is a debate about when to apply fertilizers. I found this actually difficult to sort of settle in the literature. And that debate's actually been going on since the 70s, and it's still going on today. Here I grabbed a 2022 review paper. They did a 3-year study. These scientists said that there's no evidence of an increase in winter injury after late fall nitrogen application. And then more recently, late fall applications have been linked with lower rates of crown hydration, which is good news. But the bad news is it increased the rates of fungal pathogens. Bad news. Other research also suggests that nitrogen is leaching after late fall nitrogen. And those authors suggested that this could be because of the cooler temperatures. There's a reduction in plant uptake of the nitrogen, and the bacteria are just not working as hard because it's too cold. So Dr. A.J. Koski, then at Ohio State, suggested that it's the type of fertilizer that you can use. And so he advocates for late fall nitrogen, but he says that you need to use bioavailable forms. So his suggestion was microbial dependent nitrogen fertilizers are best mid-fall, but bioavailable ones are best for late fall in colder temperatures. But again, we're still dealing with the possibility of increasing fungal pathogens. So there's lots to consider. Research done in 2016 showed that nitrogen fertilizers, when they were combined with beneficial bacteria, significantly increased the growth of ryegrass and tall fescue in all seasons. So they postulated that these benefits were suited to recovery after periods of stress. So it could be that beneficial bacteria are sort of the thing that's going to help mitigate the problems of when to put nitrogen. So if you don't want to put it in late fall and it's not working, then beneficial bacteria is definitely something that you can use instead. So what is the nitrogen gap? So I did a little bit of research here on how grasses deal with nitrogen in the spring. And what I found was an interesting 2017 study that showed that they showed that grasses with rhizomes, so things like Kentucky bluegrass, used fall stored nitrogen exclusively for the first 10 to 14 days of spring. So what this suggests is that early spring, and probably you all know this anyway, early spring nitrogen fertilization is less effective. In 2012, another group also showed that cool season grasses that didn't uptake nitrogen, they didn't uptake it until late spring compared to all other plant types. So it seems that grasses sort of use all the stored nitrogen from the previous season in the first few weeks of spring, and fertilizers are less effective. And then this same 2017 study found that there was only a 13% uptake of fertilizer in the new roots that were growing during the first 2 weeks, and all the old roots were being depleted and all that nitrogen used. So the takeaway here is that fertilizers are less effective in the beginning of spring. However, the problem is that nitrogen will then rapidly decline around day 10 to 14. And of course, the days will depend on your grass type and your climate. And so something's needed at the point of transition from last year's storage depletion. And of course, again, we don't want to encourage fungal pathogens. So I'm sure you know what I'm getting at. How are we going to fill that nitrogen gap? So since nitrogen is a limiting nutrient, this is really important. But what we can do is we can use nitrogen fixing bacteria. So most inoculants will utilize nitrogen fixing bacteria, not all. And these can bridge the gap by extending nitrogen fertilizers. So even though reducing nitrogen fertilizers can be cost effective, that's sort of not the main reason for reducing it. The main reason for reducing it is to reduce your fungal pathogens. And so in order to extend that sort of time of using fertilizers, you can add nitrogen fixing bacteria. Here in 2020, that's what I'm seeing here. There's a study here where I'm not going to go over everything here, but they had a reduced nitrogen application, about 50%, and they extended the time between applications. And you can see here A versus B means that there was a difference between the inoculated grass, which is here in blue, and the non-inoculated grass. And this time frame is important, that it's around October. And again, the same thing over here around October. That means that the inoculated grass had an increase in nitrogen. Green color tends to represent chlorophyll, which is directly linked to nitrogen use. So if you're getting an increase in nitrogen in the fall with plant growth promoters while reducing your nitrogen, you're also going to get a reduction in those fungal pathogens. So this is really good news. So again, there's a lot of other reasons for combining plant growth promoters. So that's what this PGPB here, this is sort of what you'll see in the literature. Sometimes it's PGPR. And you combine these with fertilizers and you can get optimal results. So one group found that it reduced nitrogen leaching. And so that's an interesting effect, because we know that in early spring, a lot of the nitrogen applications are leached through. And this can also happen in the fall. And another group found that adding plant growth promoters at 50% fertilizer rates match the effects of full fertilizers. So I actually found this in the literature in quite a few studies. And so this is just an example here. You can see that everything that says A means they're the same. So this is the 50% fertilizer rate. And this is the inoculated samples. And here you can see at 100% rate, it matches that rate. So you can effectively reduce your fertilizers to 50% when adding plant growth promoting bacteria and get the same results. So this is really important, and they can help you, again, to have a healthier both soil and plant microbiome. Okay, so now we're going to talk about microbial dead zones. So it's kind of a scary topic. So if you haven't fallen asleep yet, then this will wake you up because this is very scary. So the main thing I want you to know is that red equals dead. Okay, so if you see red, this is dead. So I found a lot of research about the effects of fertilizers. So fertilizers are obviously necessary for your grass, really important for nitrogen and phosphorus absorption. However, too much is going to provide a nitrogen to pathogens. So in 2021, they found that nitrogen was being used by pathogens more, and they found it directly linked. And this is especially true in the fall. So this is really important information. And then in 2020, this study here that I'm showing you, they found that nitrogen fertilizers, especially in low water supply, can cause these microbial dead zones in soils. So this relates to the soil disease suppression, where soils can't suppress pathogens as well. And one of the reasons may be because of fertilizers. So here you can see at the beginning, this is 0 minutes where they first added it. This is in low water concentration, and this is high water concentration. And then as time passes, you see more and more bacteria are dying. So here, all these red zones are dead bacteria. And when you have 70% saturation of water, you get less of that. But of course, we can't always guarantee that the grass is going to get 70% saturated. Even in 30% saturation, you had fully dead zones. So this is really important. And this effect was also found in farmlands. And again, we found that soil disease suppression, again, is reduced due to fertilizers. So, okay, so now that we've talked about fertilizers, we know that we can combine plant growth promoters with them. We also are interested in resistance. So we want to reduce the pathogenic fungi, not just by reducing the fertilizers, but because of plant growth promoting bacteria. So a study in 2023 found that creeping bentgrass showed 50% reduction in dollar spot when it was inoculated. And this was, what was interesting about this one is that in vitro, meaning the bacteria itself wasn't able to resist the fungal pathogen, but it looked like it was initiating plant disease resistance. So it was actually only effective when it was inoculated in the grass. So it triggered the grass to resist that fungal pathogen. And then in 2009, there was an interesting study here. I'm showing a picture here where various molds and fungi were suppressed, and specifically gray snow mold. For our purposes, that's important, in creeping bentgrass and Kentucky bluegrass. And these Pseudomonas species, the type of bacteria, beneficial bacteria, also reduce dollar spot, brown patch, and yellow patch. So you can see here, this is brown patch, and then you can see the inoculated grass, resisting that brown patch really well. And then the other thing I thought you might be interested in, this little bit of an aside, is that Pseudomonas species are gaining notoriety in research as being effective against nematodes. So in 2018, there was a study that showed Pseudomonas species were antagonistic to root knot nematodes. So here you can see the infected grass. This is the control, and this is the inoculated roots. So here you have much longer roots, and looks like they're competing actually for iron. And of course, the beneficial bacteria seem to win that game and reduce the population of root knot nematodes. So that's an additional benefit. Okay, so this is the last slide here with lots of information. The last thing I wanted to discuss was overseeding applications. In the literature, I saw that there was a lot of recommendations to do as early as possible. You want to get that seed in the grass as early as possible so that you can get that establishment. And of course, that depends on soil temperature as well. And you're the one to best decide when you want to apply that. But the concern in some of the studies were if there was an active fungal infection in the grass, Of course, that would attack young germinated seedlings. But the good news is that beneficial bacteria have been used for a long time in order to stimulate seed germination. So they produce, again, those phytochemicals, and they accelerate seedling and root formation as well as biomass. And here I just wanted to show you a study done in 2008. Again, they used fluorescent Pseudomonas. And the reason they use fluorescent Pseudomonas is because Pseudomonas species tends to be associated with most plant microbiomes. And also it's been found in many species of grasses. So that's why there's a lot of highlights about that particular type of bacteria. So in this particular experiment here, you see that it was sprayed here, and then they compared it to application to seed. And then Celeste here is a type of fungicide, and these are the controls. They did it in 2 different seasons. So they did it in the fall and in the spring. And so in the fall, they applied seed, and you see in the control, the growth in the autumn was higher than the spring, which you expect. And of course, the fungicide reduced the mold and helped increase that growth. And what was interesting is that when the beneficial bacteria were applied, there was a 96% rate of success in the spring compared to a 64% increase only in the non-treated plots. And what was interesting is there was no difference in how you applied the beneficial bacteria. You could either apply it to your seeds or you could apply it to spray. Most golf superintendents, I think, would prefer a spray. It's much easier, and that's sort of the general way that you're spraying most of your applications now. So that was also an interesting finding. So the takeaway for this is that the strategy for successful early overseeding can be by following that overseeding with inoculant spray in order to get that improved. So whether you do it in the fall, whether you do it in the spring, you should see a lot more success in germination as well as reducing snow mold. And in this particular study, those fields were infected with snow mold, so they still got that 96% rate. So that's pretty much all of the literature that I wanted to share with you today. So just in summary, what is it that I was trying to get across? So we know that Winterkill affects grass by increasing fungal disease and damage. So these are the 2 sort of main things. We have abiotic and biotic stress, and you also get this desiccation, which is a drought stress, and of course, crown hydration. Once that happens, probably the best thing for you to do is to do overseeded to overseed those dead areas. And then we know that nitrogen fertilizer is essential, but we also know that it increases pathogenic fungi. So the balancing act with nitrogen fertilizers is the key, and adding plant growth promoting bacteria can help to improve those microbiomes. It can reduce that fungal pressure, so that fungal pathogen pressure, and it increase soil disease suppression by, you're not going to get those microbial dead zones, and you're going to be able to improve your plant microbiome. And it fills that nitrogen gap without encouraging snow molds. So instead of increasing the nitrogen to increase growth, you can decrease the nitrogen a little bit and then add your plant growth promoting bacteria with it, and then have a better success. We know that beneficial bacteria produce phytohormones, something that other amendments can't do, and it reduces drought stress. It speeds healthy root growth, which is the key for any kind of stress, including the transition from winter to spring. And it will facilitate better green up, both when applied in the fall and in the spring and throughout the season. And lastly, if you apply it early on overseeded grass, even if you're dealing with fungal infections, you will see an increase in germination rates in order to have a better and healthier grass. So that's everything I have for you today, and I really appreciate you taking the time to listen to me.